I'm very pleased tonight to have our esteemed guests from Finland, Professor Vesa, and now is the challenger, right? Kiviniemi, professor
That's
for.
good.
That's
Wow.
good.
Yeah,
Okay.
yeah, perfect.
Thank you. I'm sure it wasn't. Um,
Close to it anyways. Yeah.
Professor for functional neuroimaging in Oulu.
Yeah.
And he'll be talking about ultrafast MREG imaging and the glymphatic solute driving mechanisms. Vesa is one of the pioneers of resting state MRI and leads our fMRI research group at the OFNI. You're a neuroradiologist as well, and the maybe one of the first guys in Europe who worked on this functional resting state MRI, if I think if I know it
Yeah.
right.
Yeah.
So you collaborated with Bharat Vishwal
Yeah.
in Milwaukee in the early 2000s, and you separated the functional brain areas, independent component analysis, I believe, as well.
Yeah. That's it. Yeah.
And right now you're developing multimodal brain imaging, ultra-fast MREG sequences on the 3 Tesla Siemens. You just told me as well that you're looking more into physiological pulsation imaging, which maybe we'll
Yes.
see today as well, and guided therapy.
Yes, indeed.
Um, without further ado, I'll leave the stage to you. Thanks for being with us tonight. And we're excited to hear about your research.
Okay, guten Abend, meine Damen und Herren. Mein Deutsch ist nicht so gut, so ich muss switch to English quite quickly, but I did try to learn 2 years of German when I was a kid in pre-high school. So that's it a long time ago. But yeah, so I'm gonna talk a little bit about ultrafast imaging, and then I'll try to minimize these off. Yes. And ultrafast brain imaging, and then a little bit about this FUS. We got 3 million euro donations, and we got the focused ultrasound to our clinic as well. So this is my group, and then Mike Nedergaard popped by. She's the inventor of the glymphatic brain clearance system. So we are in close collaboration with her. I try to put the thanks first because of the I usually don't get into the end of my talk and then I forget this. So here is Maiken again. She is our honorary doctorate. This Sir Peter Radcliffe, a Nobel Prize laureate of the hypoxia-induced factor, and this is a dean of biomedics here in biocenter here in Oulu. Actually, my classmate from medical school, Peppi, we used to be the pair in our studies, and she's collaborating with Sir Peter Radcliffe. This is Kullervo Hynynen. He's the pioneer of the focused ultrasound. Has a small research center in Sunnybrook, only 250 million Canadian dollars there worth for research. I don't know if anybody else having that much of money to do research. Jürgen Henning, you know very well. I'm a big fan of his great work and using it every day. And then Kari Alitalo, a leading vascular researcher. David Bose as a big name in NIRS. And then Lauri and Teemu are our collaborators here. So I'd like to thank these people ahead. Sorry about the Finnish language. Actually, I should have been in English, but you might have seen this picture. The brain has this glymphatic clearance mechanism, which is using the pulsations of the arteries, or that's what they see in the mouse brain in the cortex. That the pulsations here are pushing water along the perivascular, periarterial spaces, Virchow-Robin spaces, and then the glia limitans covering the perivascular spaces, which is actually the outer edge of blood-brain barrier, has a lot of these aquaporin channels that are very vital for the lymphatic function. And the story goes that there's water being pushed into the brain, and it has been thought to be taken out of the brain in the perivenous spaces. We are not completely aware of this whole system, how it's taking place, but as you might have heard, the brain does not have the lymphatic vessels or nodes that we have from the neck down everywhere else in our body. We have the lymphatic vessels next to the arteries. But in the brain, it seems that these perivascular spaces are doing, at least in the cortex, they are taking that job and then taking stuff in and out of the cortex. So Maiken showed this talk in her talk in 2014, and she was showing that there's, as you can see, this is the mouse cortex. And in the perivascular space, you can see these green tracer molecules being moved with every heartbeat that you have here in the periarterial space. Well, it's not a big surprise that arterial spaces are dominated by cardiac pulsations, surely. If you go into the perivenous space, that's actually veins, where you have, in ways, you have the respiration dominating there. And so it's a little bit different story there. Maiken said in her talk that there has to be other forces than the cardiovascular drive. And at that time, I already had our ultrafast fMRI from Freiburg and MREG, and then I had some images, so I showed that to her. I want to talk a little bit about the latest paper she had last October, where if you have a brain trauma, they had a brain trauma in a mouse model, always the same trauma in the same side, in a piston model. And after that trauma, you start to have edema increasing. There's increasing edema in the brain as a function of time. But if you use certain drugs, you can prevent that from happening. So you have a cocktail of 3 adrenergic blockers, and you can prevent that edema from happening. And she asked that, do you know why the brain starts to have the edema? And I thought, well, there's some, extra production of CSF or something like that. No, that's not the thing. And she also measured the functionality of these mice, all sorts of maze and cognitive skills. And in long term, with that simple cocktail of noradrenergic blocking, she could make the mouse get well and better. And here you can see they put tracer into the control mice. You can see in 20, 40, 60 minutes, the tracer moving into the brain. In a trauma mouse, the glymphatic function stops. And when you use the PPA drug, it's not completely recovered, but much better than without the thing. And when I talked to our neuroanesthesiologists, they went quite pale because they are continuously using noradrenaline for brain trauma patients, because they sedate them, their blood pressure goes down, and then they put them on noradrenaline, and that causes a vasoconstriction, which is actually not very good. Usually, this is a picture of lymphatic vessel, and they've measured how quickly they move. And normally there are these vasomotor wave fluctuations that are taking the debris out of the lymphatic system. In the trauma mouse, there's nothing moving basically. And with the PPA drug, you can actually quite nicely, not completely return it, but make it much better. So I'm going to show you this picture because when I saw it the first time, this is quite astonishing. This is actually a lymphatic vessel doing slow lymphatic pulsations. You can see it getting bigger and smaller in a non-trauma control mice. Okay, you can, and here you can see the signal. Sorry, by the way, there is this line on top of this. I cannot move. Is it on? It says this meeting is being recorded. I don't know.
No, that's perfectly fine. We don't see that.
Oh, you don't see that? Okay,
No.
so it's only here. Great, brilliant. So you can see this lymphatic vessel pulsating, and it's full of the tracer molecules, and then there's something coming out of the brain. Okay, then here is the trauma mice, and you can just see that basically while there was a flow in the control mice, there's nothing happening in there's only a lot of, from the trauma, there's pain, and there's a lot of cardiac pulsatility. Respiration is faster, but nothing is moving anywhere. Then when you use the blockers PPA cocktail, please have a look what happens. Boom. So the lymphatic pulsations is actually the one that is missing in the trauma brain. And then if you can look closely here, you can see the black dots that are not here in the control, nor are they here in the trauma mouse. But in the functioning trauma mouse, you have these black dots, and those are actually debris from the trauma inside the lymphatic system. Well, this is a lymph vessel, surely, but the lymphatic system is pushing stuff somehow here. We don't know how, but that was, I have never seen these slow lymphatic pulsations. So, I mean, every time I hear her talk, she comes up with these astonishing things, and they are being published either in Science or Nature, just not to offend the other journal. That's my, take on it. It's just amazing what she's doing. So I'll try to. So the idea is that if you want to restore the brain better after trauma, you should keep up the lymphatic system with these PPA blockers of adrenaline. That was pretty amazing. So in the way lymphatic system has been researched, you put into the cisterna magna of the mouse, you put some tracer, and it gets into the cortex. And unfortunately, through age, it goes down. The number of aquaporine channel goes down, and the perivascular spaces start to get jammed. And well, through age, I think we all know that you feel it too. When you're a young punk, you can, party all night long, no problem. But in the morning, when you're older, it's not so easy anymore. Unfortunately, this might be one of the reasons, at least for mice, and I think also for humans. If you put amyloid or inulin into the tissue, the same way, through age, it gets lowered out of the system by the lymphatic clearance mechanism. So, and if you want to compare it then to the human brain, again, sorry, that's the Finnish language, but Per Eide and Geringstad in Oslo, they've done a really nice job. They've been putting, taking gadolinium into the lumbar spine, only 1 milliliter, not much, 1:1 dilution, and then they have a baseline measurement, and then they follow it through time, and you can beautifully see how the gadolinium gets into the brain, and it looks basically identical to the mouse stuff that's been imaged. One of the funny things is that nothing much happens in the white matter, and that's also something that we do not know actually that well. It's a cortical system. The other thing is that it doesn't completely get away. Gadolinium, if the gadolinium leaves the tracer molecule or the carrier molecule, sorry, it stays there. It's a 3D gadolinium 3D plus. It stays wherever it gets. It just doesn't leave anymore. That's one of the things. But also these are not healthy controls. Obviously, these are NPH, normal pressure hydrocephalus patients, so we don't actually know how this functions in healthy controls currently. I mean, I've heard and I've been reviewing some papers where they postulate that when you put into the IV, you get some tracer through the plexus into the CSF, and then that gets moved into the lymphatic system. But it's kind of hard to convince an old radiologist that it's really very well working. This is a better way, but it's then problematic to get it into healthy controls. The other side of the thing is that when you push the gadolinium into the spine, actually 2/3 of that gets absorbed before it goes over the foramen magnum here. So there's something happening to the gadolinium already inside the spinal canal. I think the spinal canal is the sewer of the lymphatic system. It's downstream from the brain when you're standing up, and it takes everything close to the kidneys and liver through the local lymphatic system, then maybe out of the nerve roots. That would be my guess. There's a lot of space and surface area. When you are not sleeping, the lymphatic system actually activates when you are sleeping normally, but if you don't sleep during the night and you measure the difference, you have gadolinium increase in your brain. Okay, that's what the IDA group beautifully showed in this study. And it's been actually replicated by Helene and Nora's paper, where they measured the amyloid with PET tracers, and they were able to show that already with one sleepless night, you have amyloid accumulations in your brain. So as an on-call doctor, it's not always fun to read this paper, particularly when these two papers are saying that if you try to replace the sleep, it might not be optimal way of you should always get your sleep, but of course we always know that that's not always the case that you can do in your life. But so yeah, sleep is very important in terms of removing the stuff from the brain. So this is the stuff we are using. We have the MREG. We have something unique also. We have a non-invasive blood pressure measurement in an fMRI system. We have developed it. It's measuring from the cardiac to the carotid, the pulse wave velocity. You can see the pulse wave velocity here. Then we have EEG. Then we have a NIRS that is measuring oxy and deoxyhemoglobin and water. And then the dextrometer monitor, which is really nice. Well, they don't make them anymore, which is a damn pity, because you have an optic cable. You can see the optic cable, which is leaving out outside the system, and it doesn't interfere with the RF field in any way. So this is a perfect measuring system. No problems with the MR imaging, except the EEG might do something. We also have the 256 channel EGI that we are also using. So this is what we got back in 2012. Yufeng Zhang student Cindy Wang popped by here. We got a algorithm from Shella Kielholz. Jakob and Pierre from Freiburg, they gave us the MREG sequence. I just walked by Pierre's poster and I said, I mean, really, you've been scanning brain 10 times per second? I said, okay, one slice, yeah, fine. He said, no, whole brain. I went like, what? The reason I have been trying to do this is these very low frequency fluctuations. I ran into them in 1997 alone here. An anesthetized kid brain was oscillating at 0.03 hertz. And I thought, you know, oh my God, how can that be? We didn't give in a stimulus and it was going like somebody had been stimulating it. And those were these waves, and all along, because our scans were so slow, I was afraid that these vasomotor waves were just aliased cardiorespiratory noise. But now that we got this sequence from these two, we were able to really see that the cardiac and respiratory pulsations, like you can see here, are completely different ballgame than the vasomotor waves, which are actually, you can look here, they are the Biswallian functional network connectivity stuff all along. So this is where I started with Bharat Biswal in MCW. Used to be a big place in fMRI. Peter Bandettini, Eric Wong, OFNI people like Bob Cox, Paul Taylor, and that kind of people. So Binder, Ted Dio, big names in those days. So so that's how I got into it. And then the next question was, what on earth are these there for? Because I had no idea. Then I heard Mike and Stock, and she said, well, there has to be other sources of pulsation. In 2014 SFN meeting in Washington, my hands started shaking. I went to her and showed that, okay, you said that there has to be something else than the cardiac. I showed her, well, have a look at this. This is the respiratory and then vasomotor waves. She had only seen these. And I said, well, the human brain is looking completely different. It's pushing stuff, water mainly, I think, into the brain and some metabolites along, electrolytes to the arteries. But as we know, the respiration is dominating in the venous side. Whenever you inhale, you empty the veins from the brain and you produce this suction actually. And then the idea is that these low vasomotor waves are just actually controlling, but not only controlling, it seems that they are quite important, like you saw in the lymphatic pulsations just a moment ago. It's actually a very strong driving force. I'm just showing you these videos. Just, I haven't published these anywhere, but I just love them. They are so beautiful. And this is the cardiac pulse of the brain, slowed down tremendously. We can see every single beat. We can measure the cardiac pulse from the brain. Jürgen's MREG is one hell of a sequence. I mean, I won't, if I have to stop using MREG, I will quit because I won't touch the slow ball ever again. It's no good. This is just brilliant. The glymphatic researchers have been asking, Vesa, what are you measuring? Well, the brain is water, and we are seeing phase changes, phase and SFP, steady state free percussion changes because of these cardiac impulses in the brain. We see them, you know, at 1 Hz frequency or faster. We can see them. And once you have an impulse here in the artery, you have an opposing T2 change here in the CSF areas and veins because of the Monro-Kelly doctrine. Arteries are pushing 7 to 10 milliliters into the confined space. We know that water doesn't give in, something else has to give in, and it's CSF flowing out, venous blood flowing out, the same amount as the arteries are putting in every single heartbeat. Okay, we can catch that. And then, as you can see, it produces this wave into the tissue as well. So I think we are measuring how the water protons are behaving because of the cardiac impulse. That would be my guess. And we are seeing that every heartbeat. So, and this is, if you want to know what is the big pulsation all along in the brain, it is this. Oh, this should be, sorry about this, is actually based on a Finnish talk. I should have taken the English version. This is brain breathing pulse. 50 subjects again, like the previous one. So this is respiratory pulsation, and it's also hitting the white matter nicely, CSF surely, and the veins, no doubt. And I'm just showing in glass brain as well at the same. FSL eyes is a beautiful tool nowadays. You can really look at your data. I think you can appreciate that these pictures are nice. So this works like a dream, no problemo. Well, you have to know what you're doing, but still there might be some problems if you don't do it exactly. The problem is that it's not a Siemens, serial sequence yet. The problem with the MREG is that the reconstruction of the images is iterative. It's not straightforward 2D FFT. It takes time. So every single brain 3D volume we gather in 100 milliseconds, it takes 2 to 3 minutes iteratively to back reconstruct the image. So it's like basically a little bit like CT images in the old days, inverse imaging. But yeah, so but if you have a computer cluster of Linux that you can use parallel, so you can do it. Okay, the IDE people, IDE group has been doing really nice job in there. And if this is a flare image, and if you look very closely, you will, the radiologist there, you should know that of course in the flare image the free water is dark. here is the sagittal sinus, and this is a little bit embarrassing, at least for me. So all along, we have had this in our faces as long as we have had 3D flare or DF images, like you call it in seeing is dark fluid. But this is not dark fluid. This is actually light fluid. And this is also light without contrast media. And you have this peculiar place where actually the contrast media accumulates. So this light blue is the previous day, and after night you still have stuff along the sagittal sinus. I call this the Ringstad Ida space because I like what the guys have been doing. And so I think so this area here, and it's been replicated, and this is epileptic subjects like 75, or I don't remember how many, the Abraham. They also published in Nature, and they were able to show that there is this space where you have, and if you go back to your 3D flare / DF images, you can actually see that in some cases you have this area. It should be black, but it is not. And I think it's protein. There's a lot of protein there or other stuff. And I think this is, well, if you ask me, I think this is the exit route of the glymphatic system in the brain. That's my favorite number one. They are saying that you get the glymphatic out of your nose. In humans, my nose is not dripping. I don't believe that. It might be the case a little bit, but not much. In mice, it is that it goes through a lot. On the other hand, people have been putting tracers here into the nose, and it does get into the brain somehow. So it's not that clear. Of course, the other route, like I said, is the spine along spinal canal, a little bit in the perineal cerebral nervous system, and the glymphatic system. The eye is also a glymphatic system, but it actually goes this way, not our way. That's what we think so far. So there is this glymphatic perivascular space around the sagittal sinus. And if you look at the respiratory pulse, it's actually funny how it ends up here in the perivenous space along the sagittal sinus. If I would stop moving my mouse here in my old video, so you can see that it's actually accumulating somehow here the last moments of the pulse. So there definitely is something along the sagittal sinus. That's my point of view. I have a good friend in Jena also, Martin Walter. He used to be working in Magdeburg, and now I think he's also in Tübingen. And he has the professorship of this guy. And I'm a huge fan of Hans Berger and his beautiful EEG. And I heard the story how he almost died and got a bad accident, and his sister sent him a telegram asking, "Are you okay?" The same day, "I had this horrible feeling that something awful happened to you. Are you okay?" With a telegram, it was 1800 something, and he started looking for these telepathic waves, and then he ran into these pulsations, rest card and vasomotor waves in the operating when they were operating the brain. And then he came up with the EEG using actually those transatlantic amplifiers for electrics. So it was a really, really interesting story. I'm a big fan of his work, and this is his building. I gave a talk showing that we can now map these pulsations in the brain in Jena, and it was amazing to go after this talk to do, and I first time ever saw these his measurements, and that was a really, really fantastic meeting back then. It was 2019 or something like that. If you want to measure these pulsations, actually you can do so. I think this is one of the big things. Normally, in the old days, the spectrum we had stopped here, 0.25. and everything from here gets aliased over here. So it's actually a miracle that we ever saw anything from the brain in the old days. But now when we are scanning 10 times per second, we can measure at 5 hertz range what's going on. And let me walk through you. So we can see the cardiac at 1 hertz, okay? The respiratory power peak you can see here at around 0.2 in this particular subject. And you can see these are real measurements. This is the vasomotor low frequency waves that are dominant in the cortex. Here you can see the hotter the color, the bigger the power, and you can see that actually the respiration is actually dominating also in the white matter, and it's quite strong in the CSF. When you look at the cardiac pulsatility, it's very strong in the CSF. So when you go into literature, Dreha Kulachevsky and Jens Fram's group, beautiful work showing exactly the same thing inside the aqueducts and fourth ventricle and the ventricle system. You have the cardiac pulse is the strong one there. But then we found something really interesting. This is a new physiological thing. We call it the CREM, cardiorespiratory envelope modulations. So when you look at these spectra, you see these funny peaks every time you have a harmonic of the heart, basically. We get 1, 2, 3, up to 4 harmonics of the heart. Okay, and then you see these twins. And I didn't understand what they were. And then Lauri one day came, he's my student. He said, do you know what these are? I said, well, maybe they are some echoes or something. He said, no, they are heterodynes. And he had this beautiful upper class English accent almost to his voice. This is a perfect science of actually amplitude modulation. So respiration is making in these hot areas especially, respiration is modulating the cardiac, and you can see these modulations also in other places, but here it is actually that respiration is modulating that one. Then we had a look, we just make a winner take all analysis to see who is the biggest power. And you can see, so we put in the cortex, the vasomotor waves in green is the big power, okay? Everywhere else, basically, except the ventricles, the respiration is the big thing. In the ventricles and arteries, of course, the cardiac pulsations are the big ones. But then when you go into the ventricles, actually, the REM modulations are also very strong. Here is the winner take all analysis. So you can see it's mostly blue light. So respiration is the big thing. In the cortex, the vasomotor bold activations are the big ones. Then, oops, you can see that here in the front and in the arteries, the cardiac pulsations obviously are the big thing. But then here in the middle of ventricles, the interaction, the modulation is the really big thing. So I mean, I don't know how to thank Jürgen. He's just one hell of a guy. I mean, I love his sequence, period. You can see things that you can only dream of. I mean, then we thought what people are asking, well, okay, you see these pulsations, how are they related to the glymphatic system? Well, the glymphatic system doubles the speed of clearance in the night. And so the way it's thought to be happening is that intercellular space increases and you have this flushing during nighttime. And when you have this increase in the space, your EEG actually goes down because the concentration of kalium goes down because there's dilution. So there are two things that can actually change when it comes to kalium. Well, I'm saying I should be saying potassium, but I think K is for kalium. Sorry. So during nighttime you have a washing of the brain, right? So, and then when you wake up, like this mouse has done, the intercellular space narrows down and you have an increase in the concentration of kalium, which actually activates the neurons automatically, right? That's old news. So the faster your rhythm, the more you have potassium, aka kalium, around the neurons. Okay. So we then checked that out. We had a look in the human brain where we have the slow delta from 0.2 to 2 hertz. So this is really slow. And actually, it just so happens that in these areas, in the EGI measurements, vasomotor, respiratory pulsations, they increase when you fall asleep. Only a little bit in the cardiac side. So we got that in J Neuroscience. Heta is defending her thesis 7th of June, so we will hear more about that then. What's the time? Oh gosh, I have been talking slowly. Well, anyways, here are some images. Because we are so fast, we can measure the speed of the respiratory wave. This is sleep. We are about to submit this. This is in Heta's thesis also. So we are measuring the direction and the magnitude of the speed. And whenever we come to this point, end of exhalation, sleep increases the speed of propagation there. Okay. So that's pretty neat, I think. And here we are doing the same, but this is just vasomotor waves, really, really slow, 20-second waves. And you can see beautiful 3D representations of where they are going and how fast. And also here at the end, we have increases in speed in the sensory motor areas, auditory, sensory motor, and a little bit in the visual, also not picking up here. But so when I think based on this, when you have when the the glymphatic system increases during nighttime, also these pulsations, resp and VLF vasomotor pulsations speed up. We have been looking how it affects. It actually it reverses the drive of water. We can measure the water with our NIRS signal. We can measure the blood-brain barrier with DCEG that Hans was using. And actually, in the awake brain, you can see it's all light blue here. The bold signal is following water and blood-brain barrier as it should. Neuronal activation first, then bold. But when you fall asleep, look. Again, along the Ide ring start space, you have this change in the signal. The order changes, it reverses. The bold becomes the driver. It starts to push the water, it seems. So we are trying to push this out as a paper soon, hopefully. I mean, I have like 3 minutes for Alzheimer, epilepsy, primary lymphoma, and then focused ultrasound. Please tell me what you want to hear. Sorry, I've been too slow.
If I would have been prepared, I could pull up a poll now and people could vote for it.
Yeah.
But that's maybe we will spend the remaining 5 minutes on the poll alone. So
yeah.
You choose. What was it? Alzheimer's? What else?
I could go to the Alzheimer, yes. Let's go through the pathology and let's talk about the FUS when we get it working. Let's do it that way. Okay. Okay. So we are doing, it's on a regulatory phase, so we don't have any permits to use it anyway. So in Alzheimer's disease, when we look at the OFNI data or our local 1.5 Tesla or 3 Tesla ultrafast, always the same story. The variance of the noise increases in the bold signal. Okay. We looked at, is it cardiac, resp, or VLF? It seems to be the cardiac, which is actually nice because it just at the same time, there was another publication showing that when you have this amyloid in the arteries, the artery starts to become irregular and have an increased noise level. When you don't have amyloid, it's pretty regular, like same as in humans, but when you have Alzheimer's, the pulsations become irregular. The coefficient of variation increases. Then we measured the speed in 3D. This Hungarian brilliant student, Zalan Raina, used optical flow analysis so we can measure the direction and magnitude of speed. When we looked at the speed, along the arterial tree, the biggest changes were exactly along this map. That was the first time my mind went like, oh God, this is a beautiful result. Then we looked at the speeds, and in the peripheral areas where you have the narrow arteries, you have speed increase. You have the amyloid there, and or something else actually can be, not only amyloid, there can be something else wrong in the, I saw a video without any amyloid and the arteries were not doing well on a mice from Oslo. Stay tuned, Oslo is coming up big with that one, I think. But then we saw something really weird, actually close to hippocampal area, which are a very important thing in Alzheimer's, as we know. The pulses were going the wrong way. And that really shook my head. I was like what? It wasn't a guess. It was 17,000 pulses we averaged, and it's not a thing. And I think the reason is that when there's something wrong in the vessel wall and the impedance changes in the vessel walls, there's a reflection of, it's a normal wave physics. There's a reflection back towards in the water that we are measuring. And it's also so funny that no matter if there is amyloid or not, or tau or not, but if your blood-brain barrier starts to leak, like in this Nature paper by Nation and Slokovich et al., once the blood-brain barrier starts to leak, your cognition goes down. And I think these are the same areas they are measuring, the hippocampal areas here. If you have impulses going the wrong way, there's going to be some trouble. Absolutely. So we got it published in Brain. That was quite nice. In epilepsy, we have seen really strong respiratory pulses, like way, like up to 10 standard deviations bigger than normal. Also, the correlation from the fourth ventricle, which is usually the best signal for the respiration in the patients with epilepsy, it's all through the brain, just crazy. And then when we measure the speeds, again, at another point of exhalation, for some reason, the difference is the other way around. It slows down for some reason in the epileptic brains. We also did the drug naive, the same ballgame. So it's not the medication. So we got it published in scientific reports just last year. Ahmed did a beautiful job. So in epilepsy, there's something jamming the system, I think. Finally, this is our pride. Brain lymphoma usually kills everyone within a few months. This is the Finnish data, and everybody was dead over the 10-year period, even though you had the bond treatment, putting the drug into the brain. Lymphoma cells are actually growing behind the intact blood-brain barrier, safe from the drugs. They are very sensitive to drugs, but the blood-brain barrier is making the treatment so difficult. Unless you go in, open the blood-brain barrier with 30-second intra-arterial mannitol, 25%. There's an osmotic tsunami, opens the blood-brain barrier, you can get drugs into the brain with 100 times concentrations. And look, progression-free survival was, if you start this first line with this treatment, it was 100% for nearly 60 months. It has dropped a bit, like to 80%, but when you compare, these people are dead, and if you take some of these people here, you still have 60%. So this is published news, so it's really, really interesting. Then we had a look in the glymphatic system, or what we think is the glymphatic pulsations, just with the CV, coefficient of variation. Very simple, standard deviation divided by the mean signal. Cannot get much more simpler than that. And we were measuring it. And the point is exactly what we suspected. Here is a flare image, and that's the most sensitive for the width of the disease usually. But if you look at the CV, you can see it's going way beyond the margins of the flare pathology. It's all over the place, basically. And those, and then Walter is a very bright student. He thought, well, it's up, and this is contrast media increase, and then diffusion. It's way beyond those usually. So he thought, well, why don't we have a look how the patients are doing? So we couldn't say all. This was the situation where we started. This is the group of brain lymphoma patients. This is healthy controls. And then we had to look, this simple measure of the whole brain CV. And he found out that these were the ones that died. So we were able to actually differentiate them with the even margin from the controls. Then we had these two subjects that we got really worried about. So that was they were looking like these are going to die. So one of these went to this group, unfortunately, and died of the brain lymphoma, and the other one had a subdural hematoma. So we were able to actually predict from scans before any treatment with 100% accuracy who is going to die. So it's not that often with bolt signal you get to see the footstep of the reaper before he arrives. I mean, so with that, I would like to end. We are developing other stuff. We are developing glymphometer for measuring the glymphatic system and using the focused ultrasound to open and increase the glymphatic system. But that's maybe some other day. At this point, I would like to thank you and apologize for taking too long time.
Only you of everybody else. Thank you so much, Vesa. Great talk. No worries about the time. I think we all lost the time listening to your talk. That was fantastic.
Yeah, I mean this damn sequence from Jürgen, it's such a one hell of a thing. I mean, I've been doing over a quarter of a century bold imaging, and it's not very useful for clinics before Jürgen's. And this sequence changes the sensitivity of everything up to a signals. We just measured the signal to effect noises like 2000 when you look at the physiological effective peaks. It's just and they are not I think the other thing is that they are not aliased over each other because of the speed. So it is okay. It's a little bit difficult technology currently, but I mean you can start predicting mortality. It's the clinicians say there's no other lab, no, there's nothing you can do this with other than MREG.
Maybe it's a great example where technology really enables novel application and opens up new ways.
Yeah, I think, yeah, and I forgot to tell you that we just, we should have done this 10 years ago, but we just did our, we've tried it before, but we put water through a hole in a pineapple, and we just put water flow through the pineapple, and we could measure exactly the flow just measuring the MREG.
Yeah.
So
like the flow in the pineapple tissue.
Yeah, and you could see pulse and all that. We haven't even completely analyzed it yet, but it's just breathtaking. I just you know it's yeah. I mean,
great.
Yeah, I love the Freiburg, and I mean yes, and I have a soft spot for German things. I can't help it. Yes, it's fantastic.
You mentioned HIFU, right? And I invite questions from the audience, of course. Why not opening? And you mentioned as well great results if you open chemically the blood-brain barrier. Why not opening the blood-brain barrier with a HIFU with some temperature?
Well, to be quite honest, we would like to have the HIFU too, but we don't have the money for that. But there's one, I mean, in terms of epilepsy, there's very interesting paper. One single Japanese lady had an intractable epilepsy. I think she had it showed up in PET. I don't remember, was it hypo or hyper? Okay. Over or under metabolically active, but they tried to burn it, but they couldn't burn it high enough. So they had to stop at 47 degrees or something. But after that, she was 1 year medication free.
Okay.
She could drop out medication. She got pregnant. She got a child. So I think we don't know everything, and
Yeah.
I mean, yeah, I definitely think that exactly what you said, exactly what you
I
said.
mean, I'm the physicist, right? You're the doctor, but from what I thought,
Well,
with once,
yeah, Kullervo Hynynen is a physicist, and he comes up with quite interesting ideas of how to treat the brain. So I try to listen to people with new ideas. Yeah,
so as I said, right, I invite questions from the audience. I'll
yeah,
keep
yeah, yeah.
asking. I'll keep asking, but please quote or raise your hand. Um, the green lymph vessel that you showed in the beginning, that was
Yes,
very nice,
yes.
but where was it?
At the neck of a
Okay.
mouse. Yes, sorry, I forgot to tell. Yes, at the
Okay.
neck of the mouse here. So, and I thought my intuition would have been, you know, that it would have been respiration that is driving because it's close to the lung. No, it's the vasomotor. It's, I mean, and I even didn't know that lymph vessels have enough muscle to do that. I had no idea, and yet they are doing that. I asked it several years before, and I didn't have any answer. Actually, a Swiss guy has Steve Pro. He has beautifully shown it already in 2013 with another kind of not so obvious big videos like Mike, but the same thing basically. So there are these slow waves definitely there.
And if you do histology, you find like some muscle cells or so around the
Exactly,
vessels.
yes, indeed, and also veins have that.
Yeah. Okay.
But and I think that the big thing is that veins and lympha, they share the one thing that there's not much pressure. You know, the arteries, they have to have a lot of muscle because the heart is pumping and there's, you know, 120/80 blood pressure all the time. And but in the low pressure systems, you don't have so high pressure. So actually, you don't have to have so much muscle. So I think it's given us a whole new perspective on things. And the other thing that Maiken is always saying that please use in vivo imaging, because a lot of our pathology is relying on dead tissue samples
Sure.
where the anatomy and everything is just
Yeah.
completely distorted.
Yeah.
And the loss, already the loss of liquid pressure when the heart stops, it just everything collapses, you know.
Yeah.
So it's a different ball game when you have this thing called life going on, and when it leaves you. I think that should be the reaper steps we should be, you know, always trying to get. So
Yeah. Yeah, sure. In vivo imaging is just important. Everything changes once.
yeah.
What was um the name for this oscillation that you found? Crea or what was it? The envelope of the respiration on the
Well, yeah, well, Craig, we thought of calling it Rambo also, but that sounded a little bit naive. Respiratory amplitude modulation of brain oscillation, Rambo. But we thought CREM sounds a little bit more chic. Cardiorespiratory envelope modulation. It's just basically respiration is modulating the cardiac waves. That's just basic, very simple thing.
and you think there's a physiological motion, physiological meaning for it, or is it just the result of the physics that you have two oscillations and you have the resulting envelope?
That's a damn good question. I thought, well, this is something that only occurs in the brain, but it's not so simple at all. You know, the heart and the lung, they are oscillating on their own, which is called Traube-Hering 0.1 hertz wave. So there's another
Okay.
thing going on. So I think that this actually might be the Traube-Hering. So things happen in the thorax and things happen here as well. And I have a video out of this. I have mapped. I can just taking this blue envelope here. We can make another video like I showed, but it's looking completely different than what we saw from the respiration. So those are two independent phenomena, but this modulation. So this is actually how I forgot to tell that this is actually how radio works. AM radio, amplitude modulation. These two heterodynes are mathematical signs of amplitude modulation. So this is how you code voice into the AM radio. Basic simple explanation. Again, I say hooray to Jürgen Henning and MREG. I mean, you just don't see this in any other way. And in 3D, oh God, yes.
And 100 microseconds whole brain coverage. Do sympathetic inhibitors reduce brain waves clearing simply because they reduce blood pressure?
Brilliant question. I think it has to be involved. Yeah, has to. And it also allows the vessels to pulsate more. That's the other thing. Simply reducing blood pressure, it does. We have a paper in just this year published in January. We reduced the blood pressure a bit and we got stuff out of the glymphatic system from mice, and we have replicated that in humans. So blood pressure reduction also is involved, but it is also doing something quite interesting for the intracranial pressure. You should read our paper also in Fluids and Barriers this year. So we can increase the glymphatic efflux. So there will be ways, quite simple ways to actually increase the glymphatic clearance.
If you change the overall, if you affect the overall fluid dynamics within the body, the pressure here, pressure there.
I think normally when you are awake, you have these arteries pushing water into the brain and so on. But once you fall asleep, I think that also reduces the blood pressure, and then things start flowing the other way around. We don't know the whole picture. Like I was saying, you know, the glymphatic pictures that Maiken has here and Aide and others, you know, it's mostly in mice and in humans. It's concentrated into the cortex, not so much here in the white matter.
And we were looking. Sorry.
Yeah,
please go ahead.
You mentioned the DTI Alps. I have an allergic reaction because there people are saying that they are looking at perivascular spaces, but they're just only saying that. They are not using any information of the perivascular spaces. They do normal, nice DTI, fine. And then they say that here is Alps in these areas in the centrum semi-ovale. Yes, there might be perivascular spaces, but nobody is using any information of that there. So but I think DTI should be used, but it should be scanned much faster than we are currently using it, because you have these 3 pulsations that I've been showing: card, resp, and the vasomotor. And if you're not fast enough, they are going to screw your DTI also.
Well, maybe you get sort of an average if you're lucky.
I mean, yeah, over time you get sort of an average, and it's amazing how well it works. But if you want to know the directions and all that,
I interpret it in the way that it's just non-uniform diffusion perpendicular to the main axis of the fibers. You know, you have
yeah, that might be, and I might be too pessimistic about it. And I mean, at the end of the day, the p-values are right. There is something strange going on.
There is a preferred diffusion direction, which is unclear anatomically wise why a non-uniform diffusion. Whatever it is.
Yeah, you're right. And I mean, if we look at these pictures, why is this white matter behaving?
Honestly, we were
Yeah,
just recently looking at these pictures and we're discussing them in our glymph session. And we were
yes,
wondering as well, why does it stop at the
yeah.
cortex, the rim thingies there? We
Yeah,
couldn't really understand it, but I'm
yeah,
happy that it's
there is something else going on in here. Even Lundgaard, she did some pig studies where she could do something then glympho along the tracks, but it's not published yet. And I don't know there's something boiling out from her
mhm,
group. But yes, I mean the white matter is again a thing we don't know that much about. If I mean, and then there is this very
interestingly though, the Alps index is on the white matter, right?
yeah, yeah. It's I haven't been looking at them that much, but um yeah, it's
I think
they're
so.
mostly looking at this point here
Yes.
where you
Mhm.
tend to have those. But yeah, there's something going on there. But we I think we should be faster and also maybe smaller voxels and all that.
An issue I'm
Um,
having with these gadolinium-based studies is we're interested in basically in water, maybe in debris, but gadolinium complexes are huge molecules.
yeah,
So
yeah,
how do these, well, you said as well, which is I think completely right, gadolinium gets lost. Maybe we only see the gadolinium ions and not the complexes. I'm not so sure. But
yeah.
does aquaporin transport the huge complexes?
No, no. Hey, yeah, yeah. Um, yeah, that's a very good point. So this is a drawing, right? And
Yeah.
this is the rumors. The rumor has it that this was um um the reviewer made them to draw this first arrow here, basically. So actually, you're exactly right. These aquaporin channels are very water specific, and there was a nice review about that. It's a wonderful molecule. Inside the aquaporin 4, there is a spot in the crystallography analysis where even polar water molecules can get stuck. It's so tight. So definitely you don't have, Gadoputro going through here. Hell no. Nothing goes here except water mainly. But the thing that this where people think that how this is working is that you have these small spaces between the astrocytes. But we don't really know because it's so hard currently to image. We don't know. Also here, if you look at the latest drawings, these are always drawings. We don't see it that well.
Sure. Yeah.
This is assumptions. So unless until we can measure it with this and image it with this accuracy in live, then we know for sure. But and this is only again cortex. So it's 600 microns into the cortex, and that's it with the in vivo imaging. You don't get deeper currently. There are some other techniques where you can measure the whole brain with some other fluorescent mechanisms. Yes, MRI too in animals, but so we don't know. But these are very vital, and I think the pulsation is pushing water into the astrocyte. That's my theory, and it's going to be wrong somehow, but I don't know how it's wrong. But you're pushing water here into the astrocytes, which are swelling and narrowing. They
Mhm.
during daytime they are swelled, and somehow during nighttime they shrink. And it's well, and Maiken says again, it's the noradrenergic control of that. So it's been clearly shown that noradrenaline plays a hell of a big role in this whole system. So
So you're saying maybe the gadolinium gets through these gaps in the
yeah, yeah,
feet, okay,
yes,
of these
exactly.
cells?
So yeah, so there are some like 20 nanometer or something like that
Uh,
gaps here that stuff can go through. Yes.
the
So
leaky.
yeah. So the outer part, the glial limitans of the blood-brain barrier, where you have the perivascular spaces, that can be leaky. Yes.
And then there was maybe the last question is there's as well the study, I believe, from was it Heidelberg, where they just saw gadolinium in systemic in the blood, and after like a few hours, they found residual gadolinium somewhere in the brain.
Yeah, that's actually I'm reviewing a paper which is really strongly relying on that. And I have hard time as a radiologist, but then again, I mean, you have if you are using, there was this debate that gadolinium accumulates into the, you know, cerebellar nuclei and all that stuff. But it hasn't been shown to be dangerous, but it does accumulate and it does have to get somehow there. And I think it might be the plexus and the blood to plexus and blood to CSF interface that might be more leaky. Yeah, surely. But and maybe this, but I think here the basement membrane and the endothelium are really tight, so we don't get that much through here. If the molecules in the arteries are really, really small, I don't remember.
It was, I think, no, I mean, the original paper, I just opened it. It's from Hoffmann and Radbruch from Heidelberg. I think they just had the commercially available contrast agents.
Yeah. I mean, yes,
And
yes. I mean, but I'm saying that if you have a very small fits text run, it goes through. It's been known. So a few nanometers, it can go through here. Even this is not 100%. No. So you can have stuff going through. Yes, it's been known in mice. But like you are exactly saying, right, that the normal gadolinium contrast media, they are huge, and I don't think this is the route. But there's also a Lund group, in Sweden, northern Umeå. They have done the same thing. I've heard two talks showing the same thing, like the Heidelberg. So you have some accumulation over time into the brain, yes. And if you measure the difference, you do see stuff. So I have to admit that I have to believe that, but yeah, it's not a very big change. Yeah.
Garubutrol is what they used.
Yes, yeah.
Okay, well, maybe the very last question. This is a very there's one from the audience just coming in. What would be the mean distance between any artery and the veins in this figure? What would be the mean distance between artery and the vein in this figure? Okay.
That's what is the mean difference between penetrating artery and draining vein in the cortex? That would be the answer. Sorry, I don't remember the number now. It depends maybe on the size of the mammal too. In humans, should know. I remember that it could be half of this, but in Biswal and Hyde, they made a paper where they showed that if you had these huge draining veins in the cortex every 1.5 millimeters or something, millimeters, and if you had the voxel size around that, then you got better bold responses. So maybe it's something like 75 millimeters. I mean, you could Google that out, I think. I don't remember by heart. So that might be the difference between the space in general. But on the other hand, if you take a lump of cortex, you have those capillaries all over the place, venos, arterioles, a lot of them in the space of 1 millimeter. So MREG has a 3 by 3 by 3 millimeter voxel, so there are thousands of capillaries and all that kind of stuff there already. So a lot.
Thank you. How does the waste go out? It doesn't go through the aquaporin channels, right? How does it go out?
Yeah, I think it's the same. You can see the so that would be my guess. And then the other thing I had very hard time believing, but we have an in Yari's paper from this year, we have a picture out of that. So you can the blood vessel walls, especially in the sagittal sinus, we have a picture of that with the ICP changes. They start to make these the cellular wall makes these nanovesicles, and they go through the whole system basically.
Okay.
The lipid layers, they form these nanovesicles, microvesicles, and they go through whatever you call them, those small bubbles. That's, I think, the other way. And I think there might be just somehow a leakage through here into the bloodstream. We
It's
don't
interesting,
know
right?
that.
Yeah.
Yeah, yeah, but
Histology may find a snapshot of where those are during the process, but
yeah,
who knows?
yeah, like I was showing the lymphatic pulsations. Not even Mike knows
Yeah.
where they. I mean, and in the videos here, I tell you that they had these the black dots here. They are the
Yeah.
chunk of. So
Yeah.
how in the hell do they get there? I mean,
Yeah.
I mean, you have to have huge hole somewhere.
Yeah. But I mean, histology should catch that, right? You should have a few debris.
Yeah. Every here and there, and I mean, these are substantial size. Some of them.
Yeah.
So,
They look big.
you should be able to see those big chunks somewhere else. Yeah,
They're
definitely.
like 10 microns maybe or so, or 5
Yeah.
microns. Yeah.
Yeah. It's huge. So I mean, sorry, didn't mean to do that.
You realize I can't discuss on 4 hours. I find it super interesting.
Yeah.
But we kept you already quite a long time. Thank you so much.
Thanks. It's always a pleasure. You know, good German stuff, MREG and Siemens, so no problem.
Thank you. And hopefully we can invite you again for an update on this very interesting topic.
Yeah. Once we get our FUS rolling, so we and let's hope let's meet on meetings, and
Yeah.
I'm laudating this lymphatic meeting in Lund next June. If you're not thinking of coming, please come, because it's really nice lineup.
I'll take a note.
Yeah. Okay. Thanks a lot.
Thank you, and thanks everyone for joining us tonight. Stay tuned for the next KIS seminar, and have a good night.
Vielen Dank. Tschüss.
Tschüss.